Muscle regeneration is a fascinating and complex process, essential for maintaining muscle health and function, especially for athletes and healthcare professionals. This video will explore the step-by-step process of muscle repair and the crucial role of stem cells, particularly satellite cells, in this journey. When we engage in intense exercise, micro-tears occur in muscle fibers, initiating the repair process. The first responders are neutrophils, which arrive at the injury site to phagocytose debris and release reactive oxygen species, ROS, to clear damaged cells. Shortly after, pro-inflammatory macrophages, M1, arrive, releasing cytokines like TNF-α, IL-1β, and IL-6 to promote further inflammation and debris clearance. Muscle damage activates satellite cells, the muscle stem cells. Hepatocyte growth factor, HGF, fibroblast growth factor, FGF, and insulin-like growth factor 1, IGF-1, play crucial roles in this activation, promoting satellite cell proliferation and differentiation. Activated satellite cells proliferate and differentiate into myoblasts, guided by chemical mediators such as MyoD, MYF5, myogenin, and MRF4. Myoblasts fuse to form multinucleated myotubes, with proteins like myomaker and myomixer being essential for this fusion process. Anti-inflammatory macrophages, M2, arrive to resolve inflammation and promote tissue repair. IL-10 and TGF-beta are key mediators in this phase. Fibroblasts produce and remodel ECM components to support newly formed muscle fibers. Matrix metalloproteinases, MMPs, and tissue inhibitors of metalloproteinases, TIMPs, regulate this remodeling process. Myotubes mature into functional muscle fibers. IGF-1 continues to promote protein synthesis and muscle growth, while VEGF ensures an adequate blood supply to the regenerating tissue. Muscle fibers increase in size, enhancing muscle strength through the PI3K/ACT/mTOR pathway activated by IGF-1. The inflammatory response resolves, excess cells undergo apoptosis, and some activated satellite cells revert to a quiescent state, ready for future muscle repair. Satellite cells are the primary stem cells involved in muscle regeneration, essential for repair and maintenance. The stem cell niche provides necessary signals for activation and differentiation, ensuring a reservoir of satellite cells for future repair needs. Research into stem cell therapy aims to enhance natural repair mechanisms, with potential applications in treating conditions like muscular dystrophy. Understanding and potentially reversing the decline of satellite cells with age is a significant area of research in regenerative medicine. The coordinated action of satellite cells and their niche is crucial for effective muscle repair and growth following exercise-induced damage. Advances in stem cell research hold promise for enhancing muscle regeneration and treating muscle-related diseases.